Hello, I'm Lucy Lawrence, the scientific content producer at Technology Networks. Welcome, because you're listening to Opinionated Science. Now, you've tuned in for an exceptionally interesting episode, in my opinion, and as you can see by the title, we'll be discussing the world of cancer immunology. Joining me today is Professor Mark Loudall from the Institute of Immunology and Transplantation at the Royal Free Hospital in London, who I am in awe of, to be honest with you. He's a true pioneer in this space. Not only will we be looking at the role the immune system plays in cancer, but we'll also be taking a look at how the immune system can be exploited therapeutically. On top of this, you'll also hear truly fascinating stories from throughout Mark's career and how he got to be where he is today. And before we start this episode, I wanted to let you know that this episode is very kindly sponsored by a leader in the space, Sinobiological. Sinobiological is an internationally recognized reagent supplier and contract research organization specializing in recombinant protein production and antibody development. They offer a comprehensive portfolio of high-quality bioreagents for immunotherapy targets. I am thrilled to say that you're about to hear my interview with Professor Mark Loudall in full. Hi, Mark. How are you?
I'm very well, Lucy. Thanks for asking.
Perfect. Firstly, it's actually only right that I thank you for taking the time out of your schedule to speak with us, because as a professor of cell and tissue therapy at UCL and the director of cellular therapy at the Royal Free London NHS Foundation Trust, and also as a pioneer in the manufacturing of cell and gene therapies, your time is more than precious. So I really appreciate you taking the time. And along those lines, I was wondering if you can tell our listeners a little bit more about yourself, how you got to your position today and your research background.
Yeah, okay. So I've had a rather peripatetic career, I suppose. I trained as an immunologist, as a diagnostic immunopathologist in London, and did my PhD looking at, actually, given today's environment, rather fortuitously mapping the immune response to viral infection, which has now come around to bite me, I guess. It does make it very frustrating when you listen to people that know nothing about it talking about it on television.
I can imagine.
And so from that, I started to be interested in how you might adoptively transfer immune cells to treat viral infection back in the 1980s. And that mutated into how you might use cells to treat firstly leukemia and then solid cancer. The first one that we looked at back in the late 1980s was renal cell cancer. And then I moved, I was sort of headhunted for a post, my current post really, at the Royal Free Hospital in UCL, to replace someone who was very famous and went on to become even more famous, a guy called Malcolm Branner. And so I replaced him in 1984, and I've been in that post really since 1984, developing it into what it is now. So, I had some ideas back in the 1990s about how we could treat patients with cancer immunotherapeutically. And lots of my colleagues at UCL are far smarter than me and have similar ideas, if not better ideas, some of which I'll talk about today. And when it became apparent that these cell therapies were going to be regulated as medicines, we were a bit ahead of the game, and we were the first centre in the UK to get a manufacturing license to make these cells as medicines. And that put us in a really good place to be where we are now, which is the largest manufacturing site of cell and gene therapies in Europe, in terms of number, not in terms of space. And with a really dedicated team. I think there's probably over 40 people in the end working currently within the Centre for Cell and Gene Therapy at the Royal Free. And we support trials all over the world, both academic and commercial. And I suppose that's the next big thing. This field really took off as people saw the opportunity to use cell and gene therapies to change clinical practice, to make them regular medicines. And obviously we were in a good place there to help companies develop. I've spun out a few companies myself, and one of which, well, three of which aren't still in operation, two are IPO'd. So I'm the chief scientific officer of a company called Immune Bio, and the co-founder of another one, Achilles Therapeutics, in this immunotherapy space. So I guess what that means is that as a centre, we support all sorts of trials in all sorts of immunotherapies and gene therapies, and it means that we get experience across a array of different types of cellular and genetic medicines.
Fantastic. So, as we know, cellular immunotherapy is a treatment approach that harnesses the body's own immune system to fight cancers. But can you talk us through the main types of cellular immunotherapies and their benefits?
Yeah, definitely. I think, I mean, we could go on all day here, so stop me if I get a bit boring. But basically, everybody on this podcast probably knows that the immune system is divided into innate immunity and acquired immunity. Innate immunity is the immune system that we have, that we share with all other animals on the planet. So even earthworms and sponges have an innate immune response, which is extremely similar to ours. And in fact, some of the cells involved in that innate immune response in sponges and leeches and earthworms carry the same molecules on their surface that our innate immune cells carry. And I've been interested in the innate immune response for a very, very long time. The other half of the immune response is the acquired immune response, and only really higher vertebrates have an acquired immune response, and that's the T cell response that you hear so much about in the current COVID world. So T cell immunity is basically the immunity that vaccines work with, and innate immunity is your first line defense. And we know that patients with poor innate immunity, or individuals with poor innate immunity, are at much higher risk of developing any kind of cancer. There's a lovely paper published in The Lancet in 2000 showing that. And so quite often it's the amount of the innate immune, or the degree of innate immune response cells within a tumor is actually prognostic for outcome. So one of the things that I've been working on most of my career is how we can make that innate immune response better. And we can talk a bit about that later. But if you think about the immune system being divided into innate versus acquired, and there are lots of different types of immune cell, which we can talk about within those two areas. But within that, you've got immunotherapies that are based around using the patient's own cells, which are called autologous immunotherapies. And then you've got immune therapies which involve taking immune cells from healthy donors and using those, and those are called allogeneic immunotherapies. And within those two, you've got both autologous and allogeneic immunotherapies of the acquired and the innate immune response that are either native or genetically modified in one way or another. So it's a very, very complicated field with lots of options and lots of academics and commercial companies developing all sorts of different cellular therapies in that space. And then to make it even more complex, we've got potentiating molecules that are used to make those immune cells work better in vivo. And so the very first cellular, or the first immunotherapy in cancer, was back in the mid-1980s from Steve Rosensberg's lab in Bethesda in the US at the NIH, where he used cytokine called IL-2 to rev up the immune response of patients. And cytokine immunotherapy remains central core of immunotherapy in some diseases. And then we have therapies that direct the immune response specifically to a tumor. Things called BiTEs, BiKEs, and TriKEs, and our own product within my company, Immune Bio, which is a pseudokine. It's a cellular therapy which mirrors the effect of multiple cytokines, which is why we call it a pseudokine. So it's a really wide field. It's very complicated, but it's not inexplicable. I mean, it looks really complicated, but actually it's quite rational.
Perfect. So then if we look to some of the cellular immunotherapy drugs, can you tell us more about the research and kind of the applications of them?
Yes. So as I've said, the immune response can either be from the acquired immune response or the innate, and then it can be different cells within it. So it's probably best to structure the answer to that in terms of the cells that are available. So if we think about the innate immune response, because that's the primary immune response we all have, and it's what's protecting us, it's involved in immune surveillance. One of the cells in that, or one of the families of cells in that, are called gamma delta T cells. So I said just now that T cells sit in the acquired immune response, and they do. But there are two flavors of T cells. There are cells which have a T cell receptor, which is the bit that binds the antigen and makes the T cell work, if you like. There are some that have a T cell receptor that's consisted of an alpha chain and a beta chain, and they're called alpha beta T cells, not surprisingly. There are T cells that are part of the innate immune response, which appear to be more conserved, i.e. they're present in more animals in the phylogenetic tree, which have a gamma delta T cell receptor. So instead of having alpha chain, a beta chain, they have a gamma chain, a delta chain. And there's less variation there. So the gamma delta T cells have a much more restricted number of tumor antigens that they can recognize. But fortunately, these tumor antigens are present on many types of tumor. And so it makes them quite applicable to lots of different cancers. So there are pretty much two different gamma delta T cells even. There's those which have a delta receptor called V delta 1, and those that have a delta receptor called V delta 2. And V delta 2 is really quite, of the gamma deltas, it's the most common in the peripheral blood. They're easy to manufacture. So you take blood from the patient or from a donor, and you can grow these cells out in culture with a drug called zeledronate. And they recognize microbial proteins and viral proteins. And many of these viral proteins are shared with tumors. And so the V delta 2 population is the one that's most commonly used in cancer immunotherapy. And there are lots of trials in the field, some quite interesting ones in brain cancer and even in acute myeloid leukemia recently. It's a bit too early to determine how good they're going to be. They're certainly safe, and some patients have shown remarkable responses, and other patients have shown no response at all. So I think that's the big story of immunotherapy. You get spectacular responses in some patients and little or no responses in the other. I think the big take-home message for anybody interested in cellular immunotherapy or indeed antibody-mediated immunotherapy, if you look at survival curves of these patients, the patients, if you're in a clinical trial and you've got placebo-controlled patients and patients treated with the active cellular immunotherapy drug, what you see almost in every trial is that the patients on placebo have the usual progression that you would expect to see in patients with that condition under treatment of standard of care. And after so many months or so many years, they all die. In contrast, if you look at patients treated with immunotherapies, even when there doesn't appear to be a great response, what you almost universally find is that some patients at the end of the trial and the end of the follow-up and everything else are still alive. And we get this thing called the long tail. So if you imagine a Kaplan-Meier survival curve, the immunotherapy treated patients often carry on and on and on. You might have 20% who effectively look like they've been cured, whereas in the placebo control, you just don't see that. So there's this really intriguing story somewhere in there, which tells us that the immune response, when appropriately stimulated and appropriately maintained, seems to be able to cure patients who otherwise aren't cured with conventional drugs. And we see that in some of the trials of gamma delta therapy, as some of these patients appear to have a striking response and go on for a very long time. But currently it's a small percentage in the trials that have been published. In addition, if we follow on from gamma delta T cells, the next cell that one can be sort of interested in are natural killer cells. And that's my field. I'm very interested in natural killer cell biology and how NK cells keep healthy individuals well, and how NK cells are responsible for immune surveillance. But even NK cells fall into multiple different families. So you can divide them and divide them and divide them. The first type of NK cell, and probably the most primitive, is called the invariant NK cell, and it recognizes only lipid antigens, and they're all presented by tumor cells expressing a particular molecule called CD1D. But they're very instrumental in not only targeting tumor cells directly, but in activating other NK cell families to also kill tumor cells. So the in vitro data and the animal model data suggest that these cells can be used to really enhance a diverse immune response to tumor cells. There's not been great evidence yet in clinical trials, but there are many, many clinical trials underway. For example, one spin-out company from UCLA has just been partnered by one of the big CAR T companies called Kite, with a very, very large upfront payment of, I think it was $88 million last year. So there's lots of interest in invariant NKT cells, partly because it fills a gap in the marketplace, but partly because they're relatively easy to manufacture. One of the things that I think we can talk about later on is the challenge of manufacturing these cellular drugs in the marketplace. So beyond iNKT cells, we come to my pet interest, which is natural killer cells. And NK cells, as I say, have been known about for well over 50 years. And they were largely regarded, when I was training back in the 1980s, as irritating cells that got in the way of your T-cell experiments you were trying to do. And then when I moved to the Royal Free, I was interested in T-cell immunity in bone marrow transplantation. And we knew from the early 80s that if you have a bone marrow transplant for leukemia from your sibling, so in other words, you've got leukemia, you need high dose chemotherapy to get rid of the leukemia, and then your bone marrow won't work, so you get bone marrow from your brother or your sister who's HLA matched, now increasingly from unrelated donors even who are HLA matched. Mm-hmm. We knew that it is the immune cells in that donor transplant that cure the residual leukemia. So the chemotherapy you get never gets rid of every leukemic cell. And if you give the donor bone marrow and take out all of the lymphocytes in it, the patient will relapse. So we know that you have to give the donor immune system to eradicate the rest of the disease. And in fact, it was proven in the mid-90s by a guy called Hans-Joachim Kohl in Munich, where he did give a bone marrow transplant to a number of patients and took out all of the lymphocytes. And then when they relapsed, he gave them just the lymphocytes and he cured them. Wow. So that was really the first prima facie evidence that the donor immune response in that setting is critical, and that all transplants work in that way, all bone marrow transplants work in that way. So we were interested in what sort of lymphocytes in that transplant were curative. And everybody was believing it was T cells. And so we looked at one of our patients at the Royal Free who didn't have a transplant from a sibling or a parent because she didn't have a donor. Mm-hmm. So we used her own cells. Obviously no donor immune system there, so no chance that she would be cured, but we did it to extend her life.
Of course.
And in fact, she did mount an immune response against her leukemia, which was her own cells. And when we looked at them, they were all natural killer cells. And when she finally relapsed, it was associated with her NK cells not working any longer. So we gave her a drug to make her NK cells work better, called alpha interferon, and she went back into remission. And she stayed in remission for 16 years.
Incredible.
And she came to see me in 2011 in my office at the Royal Free to say that she was now in her 80s and she had breast cancer. And she decided that she didn't want any therapy. She'd done very well, thank you. And she was just going to live out what life she'd got. Mm-hmm. But she wanted to know whether she still had an immune response against her leukemia, which was rather sweet. And we had lots of her leukemic cells still frozen in the lab. So I took a blood sample from her and we tested it, and she did. So 16 years after we'd seen that initial immune response in her against her leukemia, she still maintained it. So that made me think that, well, prior to that, I'd been thinking, and then after we treated her originally in the mid-90s, there was something about potentiating an NK cell response which could lead to therapy. And that's where we've got to. But other people have had the same idea. So, I'd never say that anything I do is unique. But there's a number of companies and a number of huge, all of which spun out from academic findings. So although I might talk about companies, it's simply because it has to work before it gets into a company. So you've got some pretty good data. So there are companies that are taking NK cells out of patients and activating them. There's companies that are taking donor NK cells from healthy donors. There's a company in the States called Nkarta, which is taking adult peripheral blood NK cells and growing them up outside the body and freezing them and then giving them in doses to patients. There are companies which are taking induced pluripotent stem cells and turning those into common lymphoid progenitors and then maturing them into NK cells and then making various different NK products out of that and treating them. And so anybody listening to the podcast who wants to follow that, companies like Fate Therapeutics, Century Therapeutics, and Shoreline Biosciences, partnered with Gilead, are doing that sort of work. And so because you've got an iPS cell line, you can obviously grow up large numbers of these NK cells and you can genetically modify them to have particular attributes. And those trials are looking very good. I think the Fate trials are definitely the furthest progressing in that. And in leukemia and myelodysplastic syndrome, they're already showing some very good results. So NK cells are very powerful. They don't recognize tumor-specific antigens. They recognize tumor-associated molecules. So there's no antigen presentation through the major histocompatibility antigens, which is what T cells recognize. NK cells recognize fundamental changes that occur on a cell that is stressed. So whether that's by viral infection or by mutation, they upregulate molecules called MIC-A and MIC-B and the ULBPs and many others. And NK cells have receptors for those molecules. So they are permanently patrolling our body. About 10 to 15% of the lymphocytes in your blood are NK cells. They're going around the body, identifying cells that have upregulation of MIC-A, MIC-B, ULBPs, and others. And some of those may be viral infected or they may be tumors, and then the NK cells kill them. And so we've been interested in how you can make them recognize those molecules better. So within ImmuBio, we have this drug called IncMune, and both in the lab and in the patient, it activates NK cells really for quite a long time. And if we take the NK cells out of the patient, they are better at killing tumor cells. And these cells are associated, the presence of these cells are associated with, in our early clinical trials, are associated with some beneficial changes in the patient. So the other area of NK cell activity is genetically modifying them to carry specific receptors for tumors, called chimeric antigen receptors. And companies like Takeda and Artiva and even a Dutch company called Glycostem are doing trials where they're genetically modifying NK cells to carry these chimeric antigen receptors, which is basically an antibody on their surface that binds specifically to a tumor antigen and therefore targets the NK cell in a different way than the NK cell receptors. So that's really a sort of brief run through innate immunotherapy. It's a huge field. It's really dynamic at the moment, something that is going to run and run, I think. We've just dealt with the innate arm. Obviously, the acquired arm is equally exciting, and certainly in many cases way ahead of the innate arm in terms of development. So the very first T-cell based immunotherapy, cellular immunotherapy, was once again from Steve Rosenberg. That name keeps popping up in the field of immunotherapy. But Steve Rosenberg in 1985 was the first man to take a tumor from a patient and grow out from it the infiltrating lymphocytes. So if you imagine you've got a tumor, the immune system is trying to kill it, and lymphocytes invade that tumor, trying to kill it. The tumor has within it lots of immunosuppressive molecules and immunosuppressive cells, which dampen down that immune response. And what Steve Rosenberg showed famously in his first trials in skin cancer was that you could grow these cells out to a large number outside the body where they weren't under the immunosuppressive response that's going on in the tumor. And you could inject them back into the patient. They would go to the tumor and resolve it. And anybody who's interested, if you go online, you'll see some of the spectacularly huge melanoma tumors on the necks of patients or on the backs of patients, just melting away over a matter of a few months because of these T cells that have been given. So tumor infiltrating lymphocytes have been around a long time. The challenge has been working out how to make them, how to give them, and certain tumors do better than others in clinical trials. And so there's a resurgent interest in this now, a number of companies generating tumor infiltrating lymphocytes products, or TIL therapy it's called. And companies like Achilles, which I co-founded with some UCL professors a few years ago, have taken it one step further and taken the TILs out of tumors and identify in the tumors specific mutations that are present only in every tumor cell. You imagine that a tumor is evolving within you if it's growing. And so some of those tumor cells share mutations and some of them have unique mutations. So if you can identify the mutations that are in every tumor cell, then you can identify potential antigens that will be made by those mutations. And then you can make those antigens artificially and generate and expand the TIL cells that you've taken out of the TIL. You can expand them specifically against those antigens. So you end up with a TIL product that can kill every tumor cell rather than one that kills some tumor cells and not all of them. So the TIL cell therapy is moving on dramatically. There's 3 companies in the world that are developing TIL cells that I'm aware of, and there's probably many more. And those data are now showing good results in clinical trials, both academically and commercially. So I suppose the only other T cell therapy really to talk about is the one that has changed clinical practice. And that's why I wanted to finish on it. So what's driven this whole field over the last few years was a fundamental discovery back in the late 2000s, or the early 2000s actually, where with the invention of what's called chimeric antigen receptor T cells. Mm-hmm. And I alluded to CAR NK cells just now, but they are the poor brother currently of the CAR T space. CAR Ts, there are 2 licensed medicines. They've gone all the way through clinical trial, now licensed and can be prescribed globally. And they are changing clinical practice. So a chimeric antigen receptor T cell is very clever. We know that there are some tumor antigens or some antigens that are expressed on tumor cells, which are specific for that, which are highly expressed by that tumor cell. So if we start with the one that's famous and has had the greatest effect and the 2 licensed products, it's used to treat acute lymphoblastoid leukemia and a solid tumor called diffuse B-cell lymphoma. In both cases, the tumor cells express a molecule called CD19, and we can make an antibody against CD19. And that binds very strongly to cells which express CD19, which includes these tumor cells. And so what was done in the early 2000s and then commercialized in this decade was to take that antibody and generate a fusion protein which contains the antibody as the extracellular portion, and then a transmembrane domain and an intracellular portion, which is taken from the T cell receptor. So you end up with a receptor on the surface of these chimeric antigen T cells, which is half B cell receptor, an antibody, and half T cell receptor, the transmembrane and the intracellular domain. So when you put that into a viral vector, we all use lentiviruses these days, and you transduce that into a T cell from the patient. The T cells from the patient now express this chimeric antigen receptor. So it doesn't matter what their own T cell receptor is on that cell. That might be an antiviral T cell receptor. Now suddenly it's got a really, really strong receptor that's specific for a protein that's expressed on the cancer cell, in this case CD19. And so you can make those from the patient. You can go to the patient when they're in remission or when they've got low disease count, and take lots of T cells from them in a process called apheresis. And you can bring them into a lab like mine, which is a pharmaceutical manufacturing facility, and use a vector to modify the genes in that and carry in the gene for this chimeric antigen. And then a couple of days later, maybe 70 or 80% of the cells in that bag carry the chimeric antigen receptor. And so when you inject them back into the patient, they engraft the patient's own cells. So why wouldn't they? They expand and they target the tumor. And patients are cured by chimeric. Patients who are resistant to all other chemotherapy and radiotherapy are able to be cured in many cases by chimeric antigen receptor T cell therapy.
Incredible.
And it has changed the picture of immunotherapy around the world. It's a very expensive therapy. I think Novartis charged $475,000 for a dose, but you only need one dose. And in fact, Novartis won't charge you if the patient isn't cured. So I think this has radically changed cellular immunotherapy, and its impact is being felt now in terms of encouraging funding of the next generations of immunotherapy. So I think that's a pretty rapid summary of what's out there. There's plenty of other smaller cellular adoptive therapies that are being developed, but those are the major ones. Did I answer your question?
You did, absolutely. That was fantastic. And so I know, and many of our listeners will also know, that my next question is a big one, and you've touched on it slightly already, but I'm really interested to hear your thoughts on, looking at the current research out there at the moment that's available. Are there any targets that are showing particular promise in your opinion? And if so, are there any clinical results that you have in mind that you could share with us?
Yeah, that's a great question. I think it really harks back to my point about chimeric antigen receptor T cells. I think the results in CD19 positive diseases have been brilliant. Even there, we're seeing that tumors that are CD19 positive evolve in the presence of CAR T cells to shed CD19, so they become resistant to CAR T therapy. But the cells also tend to express a molecule called CD20. So colleagues of mine at UCL, a very, very smart guy called Martin Poulet, I think he was probably the first, or certainly one of the first, to generate a chimeric antigen receptor T cell that has both CD19 and CD20 targeting to avoid the evasion. And so lymphoma and B-cell leukemia are great targets and still show really, really good results. We have ourselves at UCL, funded by the Wellcome Trust, a really exciting project to deal with a severe complication or severe development in acute lymphoblastic leukemia, which is cerebral disease, when the disease transmits into the brain. And that's been very resistant to all sorts of therapies. Obviously, the amount of chemotherapy you can give to the brain is very, very low. And it's a very challenging area to target. But what we've been doing is developing a CAR T cell that can track to the brain and has the ability to be switched off. So one of the side effects of CAR T cells, tragically, is that you've got all sorts of T cells that have been transduced to express the CAR. Some of those are very pro-inflammatory T cells. And so one of the big side effects of CAR T therapy, and the one that everyone worries about, is called cytokine release syndrome, where after you've given the CAR T cells, they proliferate very rapidly because there's lots of tumor driving them to proliferate. And in doing so, some of them start to secrete large amounts of pro-inflammatory cytokines, and the patients get very, very sick indeed. In fact, cytokine release syndrome can be fatal. You certainly wouldn't, and many of the side effects are actually in the brain. So the thought of making a CAR T cell which deliberately goes to the brain is very scary. And so what Martin Poulet was able to do was insert into that construct what's called a tetracycline off gene. So when the CAR is manufactured, synthesized in the cell, so you transfect the cell to express the CAR. As the CAR is synthesized and those proteins move to the surface and embed in that membrane, at the extracellular domain, there is a cleavage site that is susceptible to tetracycline. And so the cells are permanently making chimeric antigen receptors and expressing them. But if the patient's treated with tetracycline, it should cleave that CAR as it is expressed. And certainly in the laboratory, that's what happens in animal models. So you can give these CARs to patients, or CAR T cells to patients, and if they get cerebral side effects, you can treat them with tetracycline, which is a routine antibiotic, and it should cleave the chimeric antigen receptor as it's expressed, and therefore switch off the cell. And if once you've controlled the cytokine release syndrome, you can then stop the tetracycline, and you would switch back on the chimeric antigen receptor expression. So that's a trial which we're hoping to start very soon, and we're looking at the manufacturing complexities of that. But I think those sort of things, that's just an example that I can quote because I'm able to, because it's our study. But there are very exciting similar studies being done with genetically modified cells to either switch off the signaling or switch it on again. I suppose one that's out there and known very well came out from a friend of mine, Katie Rezvani's group at the MD Anderson. And Katie's another brilliant doctor that I know, who has made NK cells from discarded or unused umbilical cord blood donations, and genetically modified those to carry the same CD19 CAR that many people use, but also to carry a gene to make a cytokine that's critical for NK survival, called IL-15. So there we have a cell which is equally genetically modified twice to have both a targeting receptor, a chimeric antigen, and IL-15 to help the cells survive. So I think that's where we're looking at in terms of really exciting things over the next few years, these multiply edited cells, particularly as CRISPR technology and TALEN technology moves further and further into clinical application. So that's what I will be looking at if I was listening to this podcast and thinking about where should I be in the next 2 or 5 years.
And then, given those promising cellular immunotherapies you've spoken about, what do you see as the greatest challenges to really making them available?
That is the sort of elephant in the room, and it's something I've been passionate about for many years. I make myself very unpopular when I say this, but I'm not frightened of being unpopular. I think that the tragedy of many of these great discoveries and great trials of all sorts of therapies, whether they be cellular or not, is that as academics, we're driven to do clinical trials or to do preclinical work and then hopefully get them into clinical trials. And we're passionate about improving things for patients. But it's very difficult then, or many of my colleagues don't see the obligation that I think they're under to develop them in such a way that they can actually scale up to change clinical practice. Now, universities want us to publish high impact papers. They don't necessarily have the skill sets, nor should they, to develop them into products. But we as academics should be helping that, and that I think is probably what I've been most busy at in my career. And so that means that from get-go, you've got to think about how you make these at scale for patients. And we've seen this problem with chimeric antigen receptor T cells. The early trials were focused on treating 6, 10, 15, 20 patients, not 10,000 patients. And so I think one of the challenges the field faces is getting a handle on how to manufacture these products, so many of which are patient specific. So the patient is involved in the manufacture of their dose, their therapy, which is just not the case for any chemotherapy. Or if you go and buy aspirin, no one says, "Oh, well, madam, you'll have to come in 10 days earlier so that we can take a blood sample
So
from
we
you."
can check, yeah.
So it's a different dynamic. It's a different world that we live in for these therapies, and so manufacturing challenges are there.
Eloquently said, Mark. What a delight it's been to spend this time going over this with you. I think your explanations are fantastic and really insightful. So thank you for this, and thank you for your work and for spending the time with us now.
Not at all. I've really enjoyed it, and people are very free to send me emails and ask me questions. This is something I'm really passionate about. Thank you for setting up the podcast, Lucy.
Thanks for listening to this episode of Opinionated Science, and a special thanks again to Cynobiological for sponsoring this episode. We hope you enjoyed listening, and if you did, make sure to subscribe so you can join us again soon.